The SciComm Collective acknowledges the Whadjuk people of the Noongar Nation as the traditional custodians of the lands we study, write and record on, and their sovereignty was never ceded. Hello and welcome to the SciComm Collective.
Different useful knowledge in different areas.
The joy is sharing it, making science accessible, the ignored final step of the scientific method.
Every different perspective.
Science is culture.
The SciComm Collective brings science and science communication to you.
Hello. Welcome to another episode of the SciComm Collective Student Podcast. I am Ray Kumar, your host for this episode. Today, we have with us in the studio Professor Neil Gemmell from the University of Otago, New Zealand. Welcome to the show. Would you like to kick off the episode by telling us a bit about yourself?
Well, thank you very much for having me. I'm a professor of genetics. I'm based at the University of Otago in New Zealand, and my main area of interest is in genetics.
So when you started collaborating with other researchers, how vast was the difference or the similarities that you felt coming from New Zealand?
Oh, look, I think the language of science is reasonably international, so, and dare I say it, Australia and New Zealand have very similar sorts of approaches and cultures to the way we work, so it's actually incredibly easy to work with my Australian colleagues.
How about when you try to reach out more to lay audiences? Is that challenging or?
Look, I think it can be challenging. I think one of the things I try to remember is how I would talk about the science to my family. So I grew up in a small urban part of New Zealand, adjacent to Wellington. Most of the people I worked with worked in industry, they worked in factories, they worked in offices. They were smart but not highly educated people. And so when I'm talking to my family about my science, I was always mindful that I needed to explain it in ways and use analogies that they were familiar with and comfortable with. You didn't want to ever talk down to people, but I wanted them to be able to understand what we did. And I also grew up in a family where I was the first person to go to university, so nobody had any idea of what university studies were like, except for what they saw on the TV. It was about explaining why research and study was important, trying to explain why things that seemed reasonably esoteric, like, for example, sex change in fish could be useful, and really why what we did could make a difference in the world, why they should pay their taxpayers dollars to support the sorts of things that we do, was an important part of the conversations we used to have. I mean, my parents are long dead now, but, those were the sorts of conversations I used to have with them and their friends on a relatively regular basis when I was starting out.
So on that note, could you tell us a brief about what you're working on?
So that's one of the questions I always dread because my interests are broad and eclectic. So I'm a geneticist, but I work on lots of different things. So I've worked on, for a long time now, on fish that change sex. So they start out life as females and become males. There are other fish that start out life as males and become females. Many people will be familiar with some of these species, like, for example, clownfish, which are famous for a movie Finding Nemo. And without shocking the audience too much, the reality of Finding Nemo, apart from the fact that fish don't talk and a number of other possibilities, Finding Nemo is biologically inaccurate. So when coral is killed by the barracuda at the beginning of the movie, what should have happened, once coral's dead, then marlin, the male, would have changed sex to female, and then Nemo, the sole surviving representative of that social group, would have been the male and would have ended up being the partner of marlin. But I have, in fairness, probably Disney isn't quite ready for that movie. And maybe the rest of the world isn't quite ready for that movie. So some fish change sex based on social environment they find themselves in, and they do that to maximise their lifetime reproductive opportunity. So marlin changing to a female would have created an opportunity for him, her, they to produce more offspring. Okay. And that social group would have survived and been maintained.
Have you told your kids this story?
Yeah, they're well aware of what Dad works on. And they're 13 and 16 now. And they think it's pretty cool. And I think it's pretty cool. I think it's fascinating that an organism, through a cue as simple as the presence or absence of a male or a female, makes a life-changing decision to alter its sexual fate. And how it does that, why it does that, we know. How it does that has been something I've been working on now for quite a while. So we know how the gonad reshapes and changes, and we also know that the change is driven by initially signals in the brain. In some instances it's about cues such as fighting, and other times it's chemicals, but most of the work that we do is socially controlled. So it's literally being able to see a male will suppress our largest females from changing sex. You remove that male, and it can take only a few days or a few hours sometimes for them to then exhibit male behaviours and then start changing sex into males. We can do this in captivity reasonably routinely now with a fish species that we find all around the coast of New Zealand. It's called New Zealand spotty, not a very attractive name, and it's actually a very nondescript fish, but its biology is amazing.
Okay.
And that's what got me into sex-changing fish. You used to catch these fish off wharves around the harbour in Wellington where I lived. It's the fish species that most New Zealand kids grow up catching because they're easy to catch. They are very aggressive, small fish with big teeth, and they're just easy to catch. So that's what most people grow up with. And so when I was reading about them, I was about 8 or 9, and I was vociferous in my reading about fish and fish biology, because that's really, marine biology was my great passion at that time. I really wanted to go and work on the Calypso with Jacques Cousteau. That was my sort of childhood dream. Yeah. And so we when I was reading about these fish and discovered they could change sex, that sort of stuck with me. And some 30 years later or something, I got an opportunity because I set up a new research lab at the University of Otago, and they gave me some money, and I thought, well, what shall I do? Shall I continue to do the sorts of things that I have done, or shall I try something new? And so the technology and opportunity and money made it possible for me to start studying sex change in fish, and I've been doing that now for about 15 years.
This is definitely a very fascinating topic, and to imagine it. So do you think you'd be able to bring, to have more conversations about it? Would it be more easier to have a proper communication part of it? Thinking about the science communication aspect of it?
For sex changing fish?
Yeah.
Oh, yeah, it's an easy story to tell.
Yeah, I mean, you can have videos about it to increase awareness. Do you get to do that a lot?
So we published quite a high-profile paper in 2019, and as part of that, we did podcasts and we did some video. We wrote a piece for The Conversation and a few other things. It's about finding the time to do it, and over the last few years, I've been doing other roles in the university, so I haven't done as much science communication as I had for a while, and now that I'm 6 weeks out of leadership, and I'm thinking that I'm going to try to write a few more popular science pieces.
Okay.
Yeah. I don't know about a book, but certainly I'm thinking actually if I could try to aim for a conversation piece, every couple of months, that would be a good target for me.
Right. So you're supervising PhD students. How do they feel? Are they more happy just doing the research in their labs, or are they more,
Yes, that's a good question. Look, I mean, the way we do research and the way we communicate research has changed quite a lot over my career. So I think, the old school way would be to go into the lab, do your research, publish your paper, and that's it, job done. But I think for those of us that are, and this is the majority of scientists in New Zealand and Australia, we are funded predominantly by the taxpayer, and I think it's beholden to us to show what the benefits are for our research. And so we have to find ways to communicate our research before we start it, while we're doing it, and then after we've done it, So what do we want to do and why? How and who is going to benefit from it? And then when we've done it, what are the benefits that have emerged from that? What is the new knowledge? How is this actually improving society? I think it's a great privilege to do what we do, to explore ideas, to find out new information. It's really, really important to maintain public funding of science and to retain people's faith in science, that we are open and honest and have these great conversations about the sorts of weird and wonderful things that we find.
Yeah.
So I enjoy that. I've talked about all sorts of different things of, obviously sex change in fish, environmental DNA, how to deal with the COVID-19 pandemic. I have opinions about many things. I have knowledge about quite a large number of things, and I try to intersect those and speak up and hopefully eloquently about the things that I think are important and I'm passionate about and that I think society needs to know about.
So with New Zealand, and obviously with incorporating
public into that conversation about science, especially since you're working with genetics. Is it tricky? How tricky would you say it would be?
Look, is it tricky? I mean, I think some forms of genetic technology have certainly public attitudes to those are different from some of the science that we do. I mean, most people don't get too upset about the idea that fish change sex naturally. But if you then said, look, I want to develop a genetically engineered fish that change sex, well, that might be a different conversation, particularly if you're dealing with a species that has that the public are interested in. So I think each conversation starts from trying to, I think it needs to be it's a two-way component communication of science. Because if you're tone deaf and you're not listening to what the audience is telling you about your science, and you continue to, blindly go on and do it, that's probably very dangerous. So my personal view is that I'm very open about what we do, and I think a couple of times we've been on the side of the fence where people are very positive about some of the things we wanted to do, and there are also others that were not so keen. So, for example, I was involved in a project in New Zealand called Predator Free 2050. So this is a project that has the ambition to remove most mammalian pests from New Zealand by 2050. And in 2017, we were talking about the idea of utilising gene drive technologies. These are technologies that would self-perpetuate a genetic element through populations, and that genetic element would make some of the animals sterile, and then ultimately you'd get population control. And, in New Zealand, as I'm sure in Australia, there are groups of people who are very, very anti-genetic modification technologies, and they are nervous about them, and they are not keen on the idea of these being utilised for any purpose, let alone one that might, but in this case, let alone one that might actually go into the environment and be unchecked potentially. And so I think that's a conversation that has to happen. I mean, we can develop technologies, but whether the public wants us to do that is something that we have to explore. And I think in a democracy you work on a consensus basis, so people are entitled to their opinions, but we, ultimately we have to follow the lead of what the general population wants. For me, I thought it was something that we should explore. I wasn't saying we'd necessarily do it, and I still think we should explore it. We must find ways to control pest species that safely and humanely and effectively, so that we can actually protect native flora and fauna that's really important to us.
How do you reckon COVID-19 and the increased public understanding of sequencing and other technologies enabled us to have slightly more in-depth conversations about genetic technologies?
So I think it's true that our Prime Minister in New Zealand, Jacinda Ardern at the time, was showing evolutionary trees of viruses and talking about clades of viruses in a relatively informed manner. And I must admit, if you'd asked me 10 years ago whether that was ever likely to be happening, I'd have said no. There has been a greater understanding of sequencing technologies, PCR, immunoassays and various other things, and also how organisms change and evolve and respond over time as a consequence of the pandemic that we're living through. It's not quite yet over, but I guess, the worst of it, I think, is probably over. And so, it does change the conversation. So when we talk about sequencing, most people are reasonably comfortable with the idea that that's sequencing the DNA or the RNA from an organism and recording its genetic code. And that genetic code can be useful for understanding the relationships among different strains or different populations of organisms or even different species. So I think that's good. So I think we're more technically au fait about some of that technology than we were. But I think you still have to start from a relatively straightforward principle because the assumption that everybody understands that is probably a wrong one. And their understanding of it may be very limited, and they may not even understand that the same sorts of processes and approaches that we've used to study COVID-19 can be applied to other species. So I'm not saying that it isn't helpful. It's actually a really good thing to lean into and explain that we're doing very similar sorts of things to what they saw on TV as we dealt with that pandemic.
I have this question. So we're learning citizen science in this semester, most of us in the masters, and we were talking about how important it is for like the participation of public in more like hard science part of it. So I just had this question that for you personally, how do you think Human Genome Project fits into it? Is it a good representation of citizen science, or is it, just data? It's a bit iffy there.
It wasn't a good representation of citizen science in any way, shape or form when we did it in, more than 20 years ago. So it was, I don't think anyone was particularly consulted about the use of their genomes. There was composite genomes. There were sequences from all around the world. I mean, it was a good international science collaboration, but in terms of the public participation in that science, it wasn't particularly great. There was quite a lot of work went into sort of the ethical, legal and social issues, and there was a big block of work that was funded by the NIH around that in the United States. But I think where we're at now is it's more participatory, where you've got people who donate their DNA and they get it sequenced, communities that have issues around heritable diseases and they want to understand why, large portions of their population are suffering from diseases in midlife, whether they be heritable cancers or what have you. And so I think we are moving into a framework now where there is it's more of a reciprocity-based approach. People have a problem, they come, scientists help solve it, and then there are benefits that emerge from that knowledge that actually helps individual families and communities. So that's a different framework from the human genome, which was just, let's find a representative genome for humans, which will then be useful down the track. But, this is part of the evolution of how we do science. And increasingly, and I was just saying this yesterday when I was having some conversations with people, is I think, we used to, do our science and then talk to people. Now, as I say, we start from a principle of trying to find community groups and understand what their problems are, and then work with them to find solutions which may include some genetics, and then we work with them to help inform whatever actions they might want to take. So with the environmental DNA, for example, it may be they just want to know what's in a stream. But knowing what's in the stream may then change their attitude to that stream and what they want to do with it down the road. So that's been quite an amazing journey because it shows you the power of information for how it can affect social change.
And how open are those conversations?
Well, they can be quite hard to start with. I mean, often it's, who within a particular area do I need to talk to? And so just finding the right people and the right forum to have those conversations can be challenging. You've just got to start. And actually word of mouth is amazingly powerful. So when you've worked positively with one group of people, often what happens is that they say, oh, this person is actually worth having a chat with. So we've done quite a lot of work on predator control with local communities. They wanted to know what's out there. They want to know that their trapping efforts are working. We can show that when they've done eradications that native species have improved in number, and the rats and other things that they're trying to control have reduced in numbers. That's quite helpful for them. We can monitor it over time. We can work with them to build frameworks that will enable them to know what's going on out there. So it's an additional piece of information that helps support what they want to do, which may be in, say, the case of New Zealand conservation, might be removing rats, stoats and possums, possibly mice, occasionally goats and wallabies.
Okay.
We've got lots of things that we don't want.
Right. So when you talk of, this is basically stakeholder analysis before you start.
I wouldn't call it, it's not quite as formal as that. But I think of it as a conversation starter. And there's only so many of these that you can do. So routinely, when we've been starting projects, we've identified groups of individuals who are already doing things. So they have a website that says, for example, we're working with a group in Taranaki on the west coast of the North Island of New Zealand. And there's a large mountain there, Taranaki Monga. It's a very sacred mountain. It's got a national park around it. It's got lots of native species there, but it's also got lots of introduced species. So what they're trying to do is to remove the introduced species so that the native species can prosper, and they want to restore the mountain back to what it once was. So they've got a website, they had funding and support, large number of iwi, so Māori tribes that were involved with this project. And so we contacted Taranaki Maunga Project, and we had a number of hui with local Māori, understood what they wanted to do, explained that maybe what we wanted to do could help them, and we just started from there. It's been a real privilege to be part of that journey with them. They've managed predator control in about a quarter of the mountain now, and there's a significant difference in what you see on the parts where the predator control has happened versus the parts that predator control is still absent.
How do you collaborate with Maoris and other local population and bring them into when you're doing your research?
Very respectfully and very slowly. So it has to be built on mutual respect and trust. And so often those early conversations are about me going in with others and the support of others. Actually, the support of other colleagues, particularly Māori colleagues, has been really important for helping to build those relationships. So none of us do these things alone. But, what I have found, and I admit that my experience may not be what everyone's experienced, is that I go in with a very genuine intent to listen and understand, and then if there are things that I can do that I think will be helpful, I'll suggest them, and then it's really up to the community as to whether they want to be involved or not. The life is short, and there are many things that I can do with my time, and I want to work with people who want to work with me and that I want to work with. So it's about assessing what that fit is in terms of the relationship. I mean, there's obviously high degrees of scepticism in some instances where people have had bad experiences with researchers or with government, and often, as a university employee, there isn't necessarily a differential between me as a university researcher and somebody coming from a government agency. And so there is some degree of scepticism. We're dealing with, in New Zealand, a couple of 100 years of colonialism, which has left a mark. Mostly when communities have been engaged with, things have been taken that they may have valued and didn't want to lose. There's always somebody wanting something. So I think it's just a question of putting your honest self out there and saying, look, we would like to do this, and here are what we think the benefits might be. And if you want to be involved, that'd be great. We'd really appreciate that. And what are the questions that you want to answer, and what can we help you with? Versus, but if you don't want to be involved, that's okay. And so I think if you take that agenda out, because if I go back 20 years, the approach would have been, hey, we want to cure leukemia, and your population would be a really good one for us to study. And there may be, not even any benefits there, but we know that we need to have, people of different ethnic origins in order for us to be able to understand, the genetic basis of these leukemias and whether they are something that happens in all human populations. Well, that's great for the researcher. What's the community getting out of that? Yeah. Possibly not a lot, except somebody else wanting to take their blood, take things that they value from them without necessarily giving back. I think I am an internal optimist. I think that the research community as a whole moved ahead a great deal, and our funders expect us to have moved ahead a great deal in terms of the way they expect us to have engaged prior to putting in grant applications and various other things.
So you talk about building trust with other communities, which is a big aspect both in New Zealand and even in Australia with the Aboriginal communities.
Very much so. And then globally. But, whether it's Indigenous communities or non-Indigenous communities, everything's about trust. Everything's about developing some level of understanding of what each person is hopeful of getting out of the relationship that you're establishing.
And science communication is a big part of it. So how do you feel about the master's project being scrapped at University of Otago? How do you think that will translate?
The reality is that the economics of universities are not great currently, and this is a global thing, and we're seeing good programs and good staff being lost across institutions across the world. Much of it is fallout from the COVID-19 pandemic. First, we didn't have a great deal of students for a while there, except for local domestic students. We had to close. There were additional costs. And then when we have opened up, we've got a cohort of students who have had a unique experience in terms of their educational journey through high school and on to university, and I am observing, and I'd be interested in your views on this, that very large numbers of them want to do anything but study. They want to go off and travel, or if they have the opportunity and means to do so, they want to go to work, they want to earn some money, they want to do something different from classical learning. And they may well come back to that journey later in life, and that would be probably a good thing, because my experience has been that students that come back to university after having done some other things come back with very clear passion and drive for what they want to do. But right now, we are seeing globally students not coming to university, and many actually have not gained university entrance. So university entrance rates in New Zealand are amongst the lowest they've been for forever at the moment.
Okay.
Last year's cohort, I think, is better, but the year before that was one of the lowest pass rates we've ever had. I don't know what the Australian experience is, but I suspect it's very similar. Universities are driven by student numbers, particularly around government funding, which most New Zealand and Australian universities are dependent on. And so therefore, if the money runs out and you don't have as much of it, then you have to stop doing something. And that means rational choices about what can we keep, what should go. It's a great shame to lose a quality program like the Master of Science Communication, but the economics of it just didn't make sense, apparently.
To add to what you were saying about learning at universities, as an international student, previously at the University of Otago, Dunedin, and now at UWA Perth, I would say that
I agree with the fact that you go out, you do a fair bit of education, then you go out, you get some life experience, and then you bring that outlook and things you have learned on the job, or even if you're traveling, you get to learn a lot of things, you meet a lot of people, you have interesting conversations, and you build a more, a repertoire of interests, and then you come back to uni, which is something that I have done. I went down genetics, and then I realized it wasn't for me because I wanted to do something, connect with people, do a lot of more networking, and now that I'm here doing science communication, I find that, okay, this has actually been my calling, because there's a huge amount of absolutely mind-blowing work that's happening in labs at the University of Otago, where my friends are there, they're doing research, and I'm like, this is brilliant, but it's not actually translating much into real life. So when you look at the field work part of it, there's a lot of gap in knowledge there, and it would be so great if we can actually have those conversations, get people involved so that they're aware of. It's not about just educating them, it's about having that conversation, having the dialogues with them, and just working more on the societal good, society justice part of it.
a key concept in science communication is storytelling and the importance of narratives. Which brings me to my next question. Could you tell us about your run-in with the fabled Loch Ness Monster?
So I've worked on sex-changing fish, I've worked on tuatara, I've worked on crows that are smart and can use tools, I've done lots of different studies of marine mammals, species of things that are important for conservation. I've figured out ways to eradicate and control pest mammal species. I've spoken about much of that research over many years. And generally what would happen is, you do publish a paper, you do a press release might get published in the local paper. Occasionally, very occasionally, you might get a bit more media attention internationally. So I remember, we did a piece of work, a couple of pieces of work that ended up in, New York Times and Washington Post and those sorts of things, and Times of London. Felt pretty good about that, right? Yeah. So I thought we were doing okay, and we've done some, very, from a scientific point of view, high quality work that ended up in good journals and has made a difference to the field. But 7 years ago, I started out on a different path, and the path was that I had engaged on social media with somebody who was known to be interested in this field of cryptozoology, so, the study of organisms that may or may not exist. And he had written a book about hunting for monsters. In this dialogue, I'd just been introduced to him, I asked him whether anyone had thought of looking for any of these cryptid species using environmental DNA. And I said, well, what about the Loch Ness Monster? If there's a biological entity in Loch Ness, then surely it's shedding DNA, and we should be able to pick up the DNA of this creature if it indeed exists. He and I both thought we were very clever and agreed that it would be something that somebody should do. And then about 6 months later, it got picked up by a reporter who wrote to me and said, "Oh, look, I saw you were having a conversation on Twitter about the Loch Ness Monster and environmental DNA. Did you ever do that project?" And I said, "No, I thought it was a good idea, but, I'm in New Zealand, I'm not in Scotland, or somebody else will do it." Mm-hmm. He said, "Oh, well, could you tell me how you would do it? Because I think my readership would be interested." And so, we exchanged a few emails, and I said, "I think you do it like this." About 6 paragraphs. Nothing particularly.
Oh, yeah, of course.
nothing particularly long. Just one of the morning emails. So I sent that off to him, and because I thought it was a fun thing to think about. Anyway, so he published this article in the National Express in Scotland, and then went. I don't know if viral's quite the right word, but it certainly went around the world. So it got picked up on the Reuters wire, and it ended up in like a whole bunch of newspapers. Your Otago scientists go search for the Loch Ness Monster, including the Otago Daily Times, our local newspaper. And then my phone really started to blow up, and the emails started to blow up, and people wanted to have interviews, and they wanted to talk to me about this hunt for the Loch Ness Monster. And I was like, I haven't actually said I'm going to do this. All I've said is that you could, and this is how you do it. So I was having this interesting internal dilemma where I was, should I lean into this and have some fun with it, or should I just go, oh no, it's good, that's just an idea and somebody will do it, but I'm not really serious about that. So I went on a couple of local radio shows, and I went on TV, and I talked to the Otago Daily Times, and I said, look, I'm only proposing this, I'm not doing it. I haven't got any money to do this, but this is what we would do, and this is how we would do it. And somebody asked me, how much do you think it costs? And I said, I think maybe $100,000. And he was like, oh, what a waste of money. I was like, well, so anyway, I went to my kids' school, and I'd been in the newspaper and on TV and that other thing, so I get this halo of kids around me. Callan's dad, we saw you in the newspaper. And I said, okay, so what do you kids think? Oh, we think it's the coolest thing ever. And so I thought, gee, look how excited they are. And I thought, if I can get 1 or 2 of these kids in every class excited about science. And then I thought, okay, I'll multiply that by the country, and then about the world. And I thought, gee, this might be the most important thing I ever do in my career. And so I said, bugger it, I'm going to do it. And so we did. And we went out to Loch Ness. And it wasn't about searching for monsters in any way, shape or form. It was really about trying to get people excited about science, excited about the new technology of environmental DNA, how we could study biodiversity literally from a cup full of water. And understand much of the biology that existed within that place. How you do science, how you establish hypotheses, what sort of controls you would need, what sort of reproducibility you'd need, all the sorts of things that I would teach in a science classroom, except I had an audience of billions of people who were reading and listening about this. So it was transformational in that way, and it was one of the greatest things I've ever done. So, as I say, we did some pretty cool science, and we might get some media attention, and there are ways to measure that using website hits and various other things. Yeah. They're right, they're tiny in comparison to the Loch Ness project, even before we'd actually done anything. I got more publicity for the Loch Ness project, for the project that I hadn't actually done, than anything I'd ever done in my entire career. and then when we actually did it, it went up. So our media people tracked this quite extensively. And after 2 years of working on this project, we'd had 15 billion estimated audience from something in the vicinity of 12,000 media stories on the project.
Wow.
So, and that includes some pretty significant things. Like I was on Good Morning America. I was on The Breakfast Show in the UK. We made a couple of documentaries. We were in the New York Times. Well, in fact, actually, it's probably quicker to say which papers we weren't in.
Yeah.
I had my 15 minutes, as Andy Warhol would say. I'd quite like to do it again, I'll be honest. It was just such fun. Lots of podcasts, lots of opportunities to talk about science. I improved as a science communicator, I think, but I had a hell of a lot of fun.
And yeah, Loch Ness, it's a topic that everybody loves.
Yeah, there's no monster, by the way. We didn't find it.
Oh, that's sad.
But I don't think anyone's disappointed about that because, as I said to people, every single project that's ever gone to Loch Ness has actually found nothing, and people still believe.
Right.
And we've been doing it for a long time now with the latest technology of the day every single time. I mean, that's one of the things that when you do a project like this, you're going to get some criticism. I think somebody from the Students' Union paper decided that it was embarrassing that the University of Otago would be associated with such a project. No respectable scientist would ever go do that. And I thought, well, actually, I'm pretty friggin' respectable scientist, actually, well known for my work, so thanks. But I pointed out that Harvard and MIT had run a project with submersibles and side-scanning sonar. I mean, a really big project in the late 70s. So they're pretty respectable universities, last time I checked.
Yeah.
So what it was is that what you get is that people take their new technologies and they go and test it at Loch Ness. Now, some people really do believe it's a monster there. That's why they go for the search. Others just want the opportunity to showcase what they're doing in an area where people give a damn.
Right.
That was me.
So how would you explain eDNA to your kids?
Well, I've done that many times. In fact, actually, my daughter stars in a Kiwi Kids video where we explain eDNA to the New Zealand primary school population. Yeah. She squirts me in the face with it. She thought that was hilarious, eDNA in your face. So the way I would normally explain environmental DNA is really quite simple, is that all organisms shed cells or small parts of their body, and for us as humans, we can think of it as the eyelashes that we see on the paper that we're writing on, from time to time, or flakes of skin, or, dare I say it, perspiration and the material that comes out that. And, of course, we all pee and poop, and kids love that. So it's all the biological detritus, the waste products that we are leaving in our physical presence. And I generally point out that when I'm in a classroom, if somebody wanted to come in, there'd be presence of Neil for a couple of days, probably, after I've been there. And then the way we collect that is mostly out of water, and I talk about using the filters as sort of molecular sieves or molecular nets. And so the kids generally have been to the pond and scooped out tadpoles with nets, or they've done other things like that. So they've got the basic idea that you can collect things by scooping things out of water, even when they're really small. And then I just point out that there's biological material that we can then sequence, and that's much easier to explain now that we've had COVID, and we can get the DNA and sequence it and relate that big databases and find what species were present in a particular sample at a particular time. So that's my sort of take-home on it, and that generally seems to go over reasonably well.
So thank you, Neil, for joining us on the SciComm Collective. Is there any other questions or anything else that you would like to add?
Oh, I don't know. I mean, I could talk for hours, as you probably can gather. I'm a university lecturer after all. That's our job. So I like talking about science, but it's been a real pleasure having an opportunity to talk to you today. Thank you.
Thank you, Neil. It was great to have you here.
Thanks for listening. Find us online at thescicommcollective.com. That's com with 2 M's. Questions or comments about today's episode? Get in touch on Instagram at the SciComm Collective. The SciComm Collective is supported by UWA's Grand Challenges Impact Grant.